All right, guys and girls, if you're watching this video, that means that you cannot do the Unit 3, Day 7 carbon transfer through snails and elodea lab yourself. That means you do not have access to a computer with Flash. If you would have access to a computer with Flash this weekend or something, please don't watch this video and instead do the lab, because what is going to happen in this video is you're going to watch me do the lab, which is obviously not as much fun as you doing the lab yourself. So I would rather you do the lab yourself, even if you have to do it this weekend. So if you won't be able to have access to a computer at any point in time and you need to watch me do the lab so you can get it done, go ahead and keep watching this video. And let's go ahead and get started. All right, so this is what the lab sheet looks like, and this is on Google Classroom, Unit 3, Day 7, Carbon Transfer through Snails and Elodea Virtual Lab. And you're going to access the lab via this link, and when you open it up, it might show you something like this, either to download Adobe Flash or to run Adobe Flash. So if it asks you to download Adobe Flash, go through the steps and download it. Don't accidentally download the other extensions, uncheck the McAfee, but you're going to have to download Adobe Flash. And those of you obviously that are on this video are not able to do that, and that's why you're here. So let's go ahead and do this. But if you somehow will have access to a computer and you just were like, I can't do this, it's giving me this error message, all you have to do is, you have to run Adobe Flash and you also have to download it. But this is...
You are setting up a snail aquarium at home. Your aquarium kit contains a bag of snail eggs on an elodea plant. The instructions say to pour the contents, including the elodea, into the aquarium, but you're not sure why you need the elodea. Why might elodea plants be important in maintaining a healthy ecosystem? In this investigation, you will use snails and elodea to explore a biological system. You will form a hypothesis about the relationship between snails and elodea, and then design an experiment to test your hypothesis.
So in this lab, we're talking about cell respiration and photosynthesis. So you're setting up a snail aquarium, and your aquarium kit contains snail eggs, right? So snails are consumers, and it contains an elodea plant. And elodea is a plant, it's telling you right off the bat, it's a producer. So it undergoes not only cell respiration, but also photosynthesis. So in this investigation, you are going to figure out, why did they, in your snail aquarium kit, include these elodea plants? So why might the elodea plants be important for maintaining a healthy ecosystem in your aquarium? And you might already have some ideas, I hope. So the way you're going to do this and fill
Explore
this out,
the lab to learn what is available for your investigation. You must click on each item in the checklist. Once you are ready to move on, click Procedure.
okay, so you're here. This part, background, we're actually going to read in a moment on the screen. This shows you how to set up the investigation, but I'm going to do all of that. But here, the introduction questions. So number 1, we're going to click problem, and then you guys are going to summarize the problem and purpose of the experiment. Problem.
You are setting up a snail aquarium at home.
And I'm sorry, I'm going to read this. You are setting up a snail aquarium at home. So this is what we just read. Sorry. You are setting up a snail aquarium at home. Your aquarium kit contains a bag of snail eggs on an elodea plant. The instructions say to pour the contents, including the elodea, into the aquarium. But you're not sure why you need the elodea. Why might elodea plants be important in maintaining a healthy ecosystem? So your problem, again, is that you received this package with the elodea and the snails. Why did it include the elodea in your package? So that is the problem.
Explore the lab to learn what is available for your investigation. You must click on each item in the checklist. Once you are ready to move on, click Procedure.
So now, after reading the background information and based on your knowledge of biology, why might the snail aquarium kit include elodea? So now you're already supposed to be thinking about it, but why might the snail aquarium kit include elodea? So We already just said, but elodea undergoes photosynthesis, right? So let's take a look at the background
All
information.
organisms are
Okay, I can't handle the robot voice. All organisms are dependent on a healthy carbon dioxide and oxygen balance. Photosynthesis and cellular respiration are key processes in maintaining this balance. Plants, through the process of photosynthesis, use energy absorbed from sunlight, water, and carbon dioxide to produce carbohydrates and oxygen. Animals and plants, through the process of cellular respiration, use oxygen and sugars to produce carbon dioxide, water, and the energy needed to maintain life. So, animals and plants, through the process of cellular respiration, use oxygen, but plants produce oxygen as a byproduct of photosynthesis. So, in an aquatic ecosystem, like in your aquarium, plants like elodea produce oxygen for the aquatic ecosystem. Plants like elodea produce oxygen for the aquatic ecosystem.
explore the lab to learn what is available.
So, that might be what you want to write for number 2. As you click through the lab setup, okay, so I'm going to go through all of these A, B, C, D, E, F, G, H, and you're going to want to just pause the video and type it in. Each one, it says write the purpose, but for a couple of these, it's just going to describe what it is. So you're just going to write exactly what pops up on the screen for each one of these items, A through H, okay? And I'm just going to go through them without flipping back and forth. Okay, let's do it. Here's our checklist now. The beaker of bromomethyl blue solution, that's this,
Beaker of bromethymol blue, BTB, solution.
is a chemical indicator used to test for the presence of carbon dioxide. This bromomethyl blue solution is green because there is already some CO2 in the solution. So that's that. That's what we're going to use to detect the presence of carbon dioxide. So this is a chemical indicator. So that might already be making you think of some chemical indicators we have used in lab this year, like the litmus in the pH lab, like the red cabbage indicator. Okay, the pond snails, which are over here.
Pond snails.
They,
Pond snails have lungs. They can breathe in or out of the water.
so these pond snails are going to be undergoing cellular respiration. Then the elodea, which is a plant.
Elodea. Elodea is an aquatic plant.
So its purpose is, to be determined, right? This is part of our experiment. But what do you think the elodea is going to do in the aquarium setup with the snails? Test tubes.
Test tubes. Use the test tubes to hold the snails and elodea.
And now, like, everything is going to be explained for how we're going to use it in the experiment. So the test tubes are going to hold the snails and elodea, and you'll see me set that up in a moment.
Grow light. Use the grow light to simulate light conditions. If you choose to leave your experiment in light conditions, you must expose the organisms to a constant light source.
So what's fun is if you guys were able to do this experiment on your own, you can choose to conduct the experiment under light, under the grow light, or under the test tube rack cover.
Test tube rack cover. Use the test tube rack cover to simulate dark conditions.
Kind of cool. The CO2 O2 cycle.
Carbon dioxide oxygen cycle poster. Use this diagram to help you write your hypothesis. Assume the cycle is true for snails and elodea.
So you can pretend this is a snail over here, and this is the elodea, and this is cell respiration, and this is photosynthesis, and this is the mitochondria, and this is the chloroplast, and the same things are true. How do oxygen and carbon dioxide cycle in an ecosystem? That's what you need there. And then this color key, this is how we interpret the results from our bromomethyl blue solution from the chemical indicator.
Color key. Use the key to determine the level of carbon dioxide in a solution.
Okay. So now we're going to open the lab notebook and we're going to follow the 7 steps indicated on screen using the procedure on page 1 to assist you. So this is the procedure over here. In case you get confused about what I'm doing, you can copy and paste even information from this sheet. This is the dependent variable, the independent variable control, and you'll see what I mean when I go and open up the lab notebook. All right, so explain how carbon dioxide cycles in aquarium water through snails and elodea. So if I place snails and elodea in a test tube, the elodea will use some of the CO2 released by the snails as a byproduct of cellular respiration, but you don't need to write all of that, for photosynthesis, decreasing the CO2 concentration in the test tube. And the way we're going to measure that, the CO2 concentration in the test tube, which is our dependent variable, the way we're going to measure that is by using that little color key, this thing down here. Decreasing the CO2 concentration in the test tube. And the way that works is because the bromomethyl blue is a chemical indicator for carbon dioxide. Okay, so now let's go back to the questions you have to answer. Oh, sorry, I keep opening this. Let's close it. If I add a snail and elodea to a test tube with bromomethyl blue, then, and that is what I wrote out. Now, how will we use our lab materials to measure the dependent variable, which is the amount of CO2? So we'll use our lab materials. We'll use the BTB, which here. Test tube and interpret our results based on the color key. Whoa, I forgot how to type just there. And this is just a general note that I need to say to the whole class, but please put your answers in a color or highlight them or make them bigger. Most of you do something like this already, but if you're writing your answer in the exact same font and color and boldness and everything as the question, sometimes when I'm grading your work, I can lose an answer or two and I'm like, oh, this person skipped a question, but really you didn't skip a question, it just ran together. There's the rest of my bioethics assignment. Okay. And just so you can see the whole answer for the hypothesis, if I add a snail and elodea to the test tube with bromomethyl blue, then the elodea will use some of the carbon dioxide released by the snails for photosynthesis, decreasing the CO2 concentration in the test tube. All right, so now we're actually going to conduct the experiment. So the cool thing about this virtual lab, and the reason why, if you're just watching this to make things easier for yourself, you should please try to go and actually do the lab yourself, because the thing that's cool and fun is that you can control the experiment yourself, but now you're going to just watch me do it. So the dependent variable is the level of CO2 in the test tubes. The operational definition, how will we measure it? So we'll measure the level of CO2 by comparing the color in the test tube to the color key. And our independent variable, we're going to decide which one we'll use. The one we're going to use is defined for us. The number of snails in each test tube. And the control, the setup of the control will be a test tube with no snails and no elodea plants, so it's just empty. A control is a setup against which the change in your dependent variable can be measured. Describe what is in the test tube used for your control condition. So because our independent variable is the number of snails in each test tube, our control condition is going to have no snails and no elodea plants. So there should be theoretically no change in that test tube. You have 8 total test tubes available for your experiment. Each of your test tubes will be filled with bromophile blue solution. We're going to use 4 test tubes in our setup. At least one of the test tubes will be used for your control condition. The other 2 test tubes will be used for your experimental conditions. Determine the setup for each tube. So again, this is defined on your handout. So this one has only the BTB solution. That one's the control. This one has 0 snails and 1 elodea plant. Test tube 2. The next test tube has 1 snail and 1 elodea plant. And the final test tube has 2 snails and 1 elodea plant. And we're going to test all of our test tubes under the grow light, so in light conditions. Oops. Okay, so now we're going to actually do our experiment.
Open the lab notebook and write a hypothesis to explain how carbon dioxide cycles in aquarium water through snails. Open the lab notebook and design an experiment to test your hypothesis. You will begin your experimental design by establishing your variables and control. Once you have answered the questions in the lab notebook, click the arrow to advance to step 3.
We already answered the questions in the lab notebook. Don't be fresh with me.
Open the lab notebook and design an experiment to test your hypothesis. You will begin your experimental design by establishing your variables and control. Once you have answered the questions in the lab notebook, click the arrow to advance to step 3.
Oh my gosh. Okay, hold on. Let me pause recording really quick and like get this to go forward because this is very silly and you guys won't be dealing with the same problem. So let me just fix this for 1 sec. I'll do that. There we go.
Begin your experiment by dragging the test tubes to the test tube racks. Drag the beaker of BTB to each test tube. Once you have filled each tube, click the arrow to advance to step 4.
Okay, so I have my 4 test tubes here, and I'm going to put bromophile blue in all 4. So they're all filled. And then in the first test tube, there's nothing, only bromophile blue. And then in the second test tube, there's no snails, but there is 1 elodea plant. Oh.
Add snails and elodea to the tubes according to your experimental design. If you want to clear the tubes and start again, click Clear Tubes. When your setup is complete, open.
So in test tube number 1, according to our experimental design, I've placed only the bromophile blue solution. In test tube 2, according to our experimental design, which is on page 1 of your lab handout, there is 0 snails, 0 snails and 1 elodea plant. In test tube 3, there's 1 snail and 1 elodea plant. And now I'm setting up our final test tube 4, with 2 snails and 1 elodea plant. So test tube number 1 is our control. Now let's take a look at what you're writing down. So in test tube number 1, the contents. Only bromophile blue solution. And what's the starting color? It's green. They're all green. And we're about to see this information laid out for us on the table, but this is how you're going to fill yours out because you're going to copy this from here. And test tube number 3, what we just set up, 1 snail and 1 elodea plant. And so because the test tube conditions are already green, we know that there is some carbon dioxide in the test tube already because bromomethyl blue is blue without the presence of carbon dioxide. So there's already some carbon dioxide in these conditions. So now let's go ahead and there's already some carbon dioxide, in these test tubes. So now let's go ahead and make predictions for each tube.
All organisms are dependent on a...
So you can see here, this is what we just set up, and now let's make our predictions. So in the first test tube, there's 0 snails, there's 0 elodea. So I'm going to predict that it's going to stay green because there's nothing in there that's modifying the level of carbon dioxide. But now this second test tube, has 0 snails and 1 elodea. So there's nothing in there that's producing excess carbon dioxide, and instead there's the elodea in there that's producing oxygen and is going to be using carbon dioxide as fuel for photosynthesis. So I'm going to predict that this test tube is going to wind up blue because it's going to have less carbon dioxide. Now test tube number 3 has 1 snail and 1 elodea. And as we were talking about the process of cell respiration and photosynthesis, remember it's a cycle that balances the levels of these gases in the atmosphere. So at the end, the test tube should still be green, relatively no change, or blue, reduced carbon dioxide, and increased oxygen. What do you think? We'll find out. Now this last one has 2 snails and only 1 elodea. So in this last one, there are theoretically 2 organisms that are producing carbon dioxide via cellular respiration, and only 1 elodea to balance their carbon dioxide output. So we can see here in our color of solution chart, there is a high carbon dioxide color indicator, which looks to me to be like a yellow shade. So that's my predicted end color for test tube number 4 that has 2 snails and only 1 elodea. So we're going to put those predictions, green, blue, green, yellow, into our lab sheet. Now let's actually carry out the experiment.
Double check your setup, then click Add Stoppers to stopper the tubes.
So our setup
if you
is
are
all
testing
good.
in light conditions, drag the rack to the grow light. If you are testing in dark conditions, drag the rack to the cover. Click Start to leave the tubes for 24 hours. Once 24 hours have passed, click the arrow to advance to step 6.
So I added stoppers to the test tubes and clicked start, and 24 hours progressed, and under light conditions, we can see, so why did we put our test tubes under light conditions? Well, it's so that the elodea would be able to undergo photosynthesis, right? In order to undergo photosynthesis, plants need a source of light energy, which is usually the sun, but in this case is a grow light. And we can see here our test tube number 1, as we predicted, is still green because there's nothing in there, right? Except for the bromomethyl blue solution, so there was no change. Test tube number 2, however, did change as we predicted. We can see here the color in test tube number 2 is blue, which indicates there's no carbon dioxide in there. And why is that? It's because the elodea was carrying out photosynthesis using up that carbon dioxide. Test tube number 3 is green still, and that's because there was 1 snail and 1 elodea, so they were balancing each other's oxygen and carbon dioxide output. No change. Test tube number 4, again, as we predicted, turned bright yellow. And why is that? That's because there were 2 snails in there producing carbon dioxide, 2 snails changing the carbon dioxide levels within the test tube.
Observe the change in color of the BTB solution in each test tube. Open the lab notebook and record the results of your experiment.
So now let's
Once you have
record our
recorded your
results.
results, click.
The end color of the first test tube was green. The second test tube was blue, which indicates, again, no carbon dioxide. The third test tube was green again because there was a balance. And the fourth test tube turned yellow because there was so much carbon dioxide being output by those two snails, and it was only balanced by one elodea.
You are now ready to analyze your data and draw your conclusions. Open the lab notebook and answer the questions.
So now you would also get the option to do a different version of the experiment, but you're not going to be able to do that because you only have access to me doing the experiment. So let's go ahead and look at your conclusion questions, but you should be able to answer all of these 7 questions on your own. So please go ahead and pause the video, and then I'll go over them generally once you're done answering the question. All right, so the discussion questions. Bromothyle blue is an indicator for carbon dioxide. Using the color key as a reference, describe how to interpret your results below. If no carbon dioxide is present in your test tube, it will be a blue color. If a medium amount of carbon dioxide is present in your test tube will be a green color. And we just saw this when we interpreted our results. If a large amount of CO2 is present in your test tube will be a yellow color, which we saw in the test tube that had 2 snails and 1 elodea. What is the relationship between snails and elodea? The snails produce carbon dioxide as a waste byproduct. Oh my God, that's my cat. Hold on. Come here, kitty. She's really into this lab. So, the snails produce carbon dioxide as a waste byproduct of cellular respiration. And the elodea use that CO2 as fuel for the process of photosynthesis. My cat can't conduct photosynthesis, but she can do cellular respiration, and she's probably doing that right now. So, number 3, analyze. Why did the color of the bromothyle blue solution change? The BTB solution changed in test tube 2 because the elodea used the CO2 to conduct photosynthesis. In test tube 4, the color changed because the snails, wow, now she wants to leave, produced excess CO2, and there was only one. Hold on 1 second, Salem. Elodea to balance the CO2 output. What was the importance of a control in your experiment? What would you conclude if the color of the solution and the control changed? So, okay, this is an easy one. Obviously, in every experiment, the control is for comparison. And then if the test tube had changed that had nothing in it, there was obviously, for example, let's say we don't know a lot about carbon dioxide. This is an indicator for some chemical we're not that familiar with. But we could determine if the color in the test tube that had nothing else in it changed, maybe that solution degrades over time. So time is a variable that you have to be mindful of. But Yeah, that there was some other variable. Or that was normal conditions, is that it would change over time, which again would mean like there's some other variable. Okay, Salem, you'll get your wish. Hold on. When you began the experiment, was there CO2 in the water? Yes. We know this because the test tubes were green. In the test tubes that contained elodea, the CO2, hold on, she's going to finish the lab with us. In the test tubes that contained elodea, the CO2 was used as fuel for photosynthesis. Which gas did the snails release? The snails released CO2. This observation is that the test tubes that had 2 snails and only 1 elodea turned yellow, meaning that they had a greater concentration of CO2 because more CO2 was released as a byproduct of cellular respiration, right? So not only do we have the observation, the color change in the test tube with 2 snails, we also have our scientific background knowledge that CO2 is a byproduct of cellular respiration that is exhaled by organisms that undergo cellular respiration, such as snails. Like it said in the beginning, snails have lungs. Based on the results of your experiment, explain why you need to add the elodea to your snail aquarium. Elodea produce oxygen for the aquatic ecosystem via photosynthesis. And that, ladies and gentlemen, before my cat cries again, is the lab. That's it. That's everything you need. That's the whole ball game, ladies and gentlemen. Have a super fantastic day. I'll see you all tomorrow on Zoom. Goodbye.